to talking about how cells take in components, whether it's through their diet or whether it's things that they create through photosynthesis, and how they change the form of the energy into a usable form that they can store and use to do cellular processes. So that's called aerobic cellular respiration. Okay, so the easiest way to think about cellular respiration is by breaking down or catabolizing macromolecules. We're going to use carbohydrates as our starting point for this process. It's not the only thing cells can catabolize. It's not the only macromolecule they can extract energy from, but it's the best example because it's the macromolecule that goes through the whole process, starting at the beginning, going all the way through to the end. Right? We'll see later how other macromolecules can enter this process at different points, but we're going to use carbohydrates as our example. Okay, so what is cellular respiration? It's the subset of metabolic reactions, chemical reactions, that cells use to get energy from food. Right? So if you're a heterotroph, if you're eating and consuming nutrients from the outside world, right, then that's things that you ingest by eating or by phagocytosis, or depending on the type of organism you are. If you are an autotroph, like a plant, then these are molecules that you have made by photosynthesis, which is what we're going to talk about next week. Okay, so we've got macromolecules that have a lot of these carbon-carbon bonds and carbon-hydrogen bonds. And through the process of cellular respiration, we're going to oxidize those bonds. And ultimately, we're going to be left with carbon dioxide. The cool thing about cellular respiration is this is done in a very stepwise, very controlled way. So it's not just lighting it on fire and releasing all the energy at once. It's very small reactions, each with their own activation energy, which each release some energy that can be stored. Right? So a cell can get approximately a third of the energy back out of that molecule. Whereas if we did it all in one step, we'd get a lot less out because a lot more would be released in unusable forms. Right? So the analogy on the left with the staircase is what cellular respiration is going to look like. Okay, so if we want to think about this in terms of the chemistry, we're going to start with some sort of molecule, and we're starting with a sugar molecule, a hexose, in this case glucose. But it's not the only thing to start with. It's just the molecule that makes this equation nice and balanced. Right? So if we start with a molecule of glucose in the presence of oxygen, we're going to release those carbons back in the form of carbon dioxide. We're going to release water as a waste product, and we're going to harvest approximately 32 ATPs in the process. Right? This number can range a little bit. It could be higher or lower. So that's an approximate value. This is going to happen in multiple steps. So in each step, a bond's going to get broken, or a bond's going to get rearranged, or a bond's going to get made. And each time we're going to transfer a little bit of energy. How are we going to do that? We're going to move electrons. So we're going to move these electrons from molecules with high energy to ones with less energy. And we're going to do some work in the process. And then each time we do that, we're going to store a little bit of that released energy. So as you can guess, there are lots of steps in this process. We're going to cover them all today, and then we're going to leave some time on Monday to go through and review, see how much you remember, see what questions you have. I'm also going to try to be very clear about what steps I want you to understand versus memorize, and what steps I just am going to present, but you don't necessarily need to know all the details of. Okay, a couple more pieces of information that you need to know before we get started, and that is this term substrate level phosphorylation. So it's going to have something to do with a substrate and an enzyme, and then phosphorylation means adding a phosphate group to something, right? So what does this mean? This means that we're going to form, in this case, ATP is our product, right? We're going to form it by moving a phosphate group to our substrate, in this case, ADP, from another compound that already had a phosphate group. So this is one way to make ATP. We can pull a phosphate off of a substrate, add it to ADP, and now we have 3 phosphates ATP. We've built a covalent bond with all that energy, and then this has a lot of potential energy to do work later. right? When we talk about the first step of cellular respiration, this substrate level phosphorylation is how we're making the ATP in that process. When we get to the end of today, we're also going to talk about producing ATP by oxidative phosphorylation. And that's a slightly different method of making ATP, and we'll learn about that a little bit later. Slightly different version of a slide I showed you on Wednesday. So here's a molecule. If it loses its electrons, and in this case, the hydrogen atoms with it, it becomes oxidized. There's got to be a coupled reaction with that. Remember, these never happen on their own. So if something gets oxidized, these go somewhere. That molecule is the one that becomes reduced, right? We're going to be talking about this molecule NAD, which can be oxidized or reduced depending on whether it's got electrons bound to it or not. So its oxidized form is called NAD+, and its reduced form, when it's carrying these two extra electrons, is called NADH. NADH can then get oxidized to go back to the NAD+ form, which can then get reduced to go back to the NADH form. So we're going to look at NAD as an electron carrier and see where it's going to get those electrons. Okay, so there are 3.5 major steps of cellular respiration. The first one's called glycolysis. So glyco for glucose-like sugar, and lysis for breaking. So we're going to break sugar in this step. There are actually 10 chemical reactions that happen in glycolysis. Your book shows you all 10. Going through and looking at all 10 may help you understand how we get from glucose to these 2,3-carbon sugars and where it is that these electrons come from and how it is we make these ATPs. But I'll show you in a minute the steps I actually want you to know. Okay, so where are we in the cell? We're in the cytoplasm. So the cool thing is this reaction can happen in prokaryotes and eukaryotes. All right, so they can break down 6-carbon sugars into 2 3-carbon molecules. By the end of this process, we're going to get 2 of these molecules called pyruvate. So that's a name you should know because we're going to follow the fate of these pyruvates. We're going to ultimately produce 2 molecules of ATP. As you'll see in a second, this is a multi-step process where we're going to spend some and then make some extra. So the net result we get out of that reaction is 2 ATPs. And then we're going to create 2 reduced forms of NAD. So we're going to get 2 NADH molecules out of this process. So think about, what did we start with? We started with glucose. What did we end with? We ended with pyruvate. All right, but we also had some things coming in and out on the sides here. So if we're going to end with ATPs, we need something. We can't make this from nothing, right? So we need ADPs and we need phosphate groups. If we're going to make 2 NADHs, we need 2 NAD+ molecules and some electrons. So ultimately, throughout this whole process, I'm going to be asking you, what are the inputs and what are the outputs of each step? And keep in mind, it's not just the starting and ending carbon-based molecules, but it's also these ADPs and ATPs and NAD+s and NADHs. And we'll get a chance to practice this in a little bit. Okay, so there are 2 major phases of glycolysis. In the first one, we're going to partially break down glucose, and we're actually going to need some ATPs to do that. So the very first thing that glycolysis does is spend some energy, which seems kind of counterproductive because the whole point of cellular respiration is to produce energy. But trust me, we're going to produce a lot more than we use. All right, so we're going to take the phosphates off of ATP, and we're going to put them on these glucose molecules temporarily, which leaves us with ADPs left over. Then what we're going to do is we're going to continue to rearrange these bonds, right? So in rearranging these bonds, we're going to not lose or gain any carbons, right? We started with 6 carbons in glucose. We're going to end with 6 carbons. So each pyruvate that's made has 3 carbons. But we're going to rearrange these bonds, and we're going to ultimately take some phosphates back off and put them on ADP. So we're going to do that 4 times. So now we've got 4 ATP formed. And we're also going to strip some of the electrons. We're going to oxidize glucose and its intermediates and put those electrons on NAD+ to make NADH. So we're going to reduce NAD. So what are our net products, right? We started with glucose, we ended with 2 pyruvates, and Again, if you follow the step-by-step diagram in your book, you'll see where these oxygens come from as well to make water. We made 4 ATP right here, but we used 2 up here. So our net output on ATP is 2. And we use some NAD pluses and some electrons to make 2 NADHs. Okay, this ATP is not 32. I told you it's 32 total. All right, we haven't made very much. We've just made a tiny bit, but we're going to make more in a little bit. And these NADHs right now are a great source of potential energy that are going to be used later in this process. So that's glycolysis. So this is the cell diagram that you guys have. This is where we're going. This is the big picture, right? So we have talked about this blue box right here on the left. Again, we're talking about in the cytoplasm. It happens in all different cell types. And we started with glucose and we ended with pyruvate. We made a little bit of ATP by this process called substrate level phosphorylation, because we took the phosphates off of something and put them on ADP. And we stuck some electrons onto NADH. Okay, great. We don't have much energy, so we need to keep going. Also, we've got a problem. We've just made a toxic compound. So pyruvate will kill your cells. So we can't end with this. We've got to keep going and do something to pyruvate to get more energy out from it and also to turn it into a form that is not deadly to your cells. Okay, so what are we going to do next? We're going to bring that pyruvate into the mitochondrion. So now we're talking about eukaryotic specific processes because we're inside this membrane-bound organelle that prokaryotes don't have. Prokaryotes do some of these next steps. They do it on their plasma membrane in a very different way. So we're going to focus on eukaryotic respiration from here on out. Okay, so we're going to, like this term says, oxidize pyruvate. We're going to strip some of its electrons off, right, and turn it into a different molecule. So here's our 3-carbon pyruvate. So we're going to do this twice for every glucose that we started with. And for each pyruvate, we're going to do a couple of things. We're going to take one of its carbons off because the molecule we end with only has 2 carbons. It's going to be released from the cell in the form of carbon dioxide, right? It's the same carbon dioxide that you have to exhale because carbon dioxide will make your cells acidic. We're also going to take some of the electrons off of pyruvate and put them on NAD+. So we're going to reduce NAD+ to make NADH. If NAD is getting reduced, that means pyruvate's getting oxidized. And then we're going to use the leftover energy from breaking this bond with this carbon to add on this molecule called coenzyme A. The resulting product is called acetyl coenzyme A, or acetyl-CoA, because it's an acetyl group hooked up to coenzyme A. So what's happened? We've lost a carbon. There's your CO2 byproduct. We've taken off some electrons and put them and made additional NADH, and we've ended with acetyl-CoA. And again, we're in the mitochondrion now. So again, to orient you, we talked about glycolysis. We just oxidized the pyruvate into acetyl-CoA. And now we're going to send it through this cycle. So we are still in the mitochondrion. And what we're going to do is see a process by which we are going to produce a little bit more ATP, again, by substrate level phosphorylation. So we're going to take phosphates off of things and put them on ADP to make ATP. And we're going to make some more electron carriers. We're going to make more of this NADH molecule that we've been seeing. And then we're going to make a cousin of it. It's called FADH2, which we'll see in a minute. Okay, so the citric acid cycle is 8 reactions. And again, your textbook will go through all 8. It will show you the structures. It will show you where every atom is and where every bond is formed or broken. If that helps you understand where these things going out and these things coming in come from, awesome. If not, stick to this version here. So we're going to, again, focus on the inputs and the outputs. Try to keep it very simple. All right, so it's a cycle. So we're going to end with something, and then we're going to start with that same something. We've got things coming into the cycle, and we've got things going out of the cycle. And again, this is happening in the mitochondria. And it's going to happen twice for every starting glucose molecule. So for every glucose we started with, we ended up with 2 acetyl-CoAs. Each one is going to go through this process. All right. The numbers in this cycle are referring to how many carbons are present at a given time. And the reason that it's tracking that is so that you can see the recycled materials. So we started with a 2-carbon molecule, acetyl-CoA, and we ended up with this 4-carbon molecule. Here's a chicken egg thing, right? So oxaloacetate was present from the previous cycle, and we're going to combine the two. So this is the first step. To power forming this bond, we're going to strip off the coenzyme A. So we added it in the previous step. Now we're going to take it off. So this was just kind of a placeholder for some energy. And we're going to make this 6-carbon molecule called citrate. In all of the other steps, what we're going to do is oxidize citrate into different intermediates, and we're going to remove some of its carbons. And all of these changes are going to allow us to extract some energy, and it's going to allow us to end up with this 4-carbon molecule that we started with in the first place so that we can do it again. All right, so if we follow citrate around, the first thing that happens is one of its carbons gets removed. So here's some more CO2 for you to exhale. So now we've got 5 carbons left over. We're going to remove another CO2 and some electrons. So in this step, we've got 2 NAD+ molecules that are going to get reduced to make 2 NADH molecules. And where do those electrons come from? They come from this intermediate that came from citrate. We're then going to take off a phosphate. We're going to smack it on this molecule called GDP. GDP is like ADP, but it's a guanine instead of an adenine. Right. This GTP molecule is ultimately going to get used to make ATP. So GTP is the direct product, ATP is the indirect product. We've still got 4 carbons. We're going to take some more electrons off it. These electrons have a little less energy than the electrons over here did. And so they can't reduce NAD+, but they can reduce this molecule called FAD to make FADH2. This is also an electron carrier. It also got reduced. It just doesn't have as much energy in its electrons. Still got 4 carbons. We're going to take off more electrons. These are higher energy electrons, so they're going to reduce NAD+ to make more NADH. And then we're going to end where we started with. We're going to bring in another 2-carbon acetyl-CoA, combine it with oxaloacetate to make citrate, and we're going to do the whole thing again. So for each glucose, we get 2 sets of these products, because we've got 2 acetyl-CoAs that we formed by oxidizing pyruvate. So for each glucose, we've made indirectly 2 more ATPs to add to the 2 we made in glycolysis. So now we're up to 4. So most of what we've made here is either waste products in the form of CO2 or these high energy electron carriers that we've reduced with these electrons. We need lots more ATP. So now we're going to follow these electron carriers and see why the heck we're spending any time or energy creating them. So make sure that you're familiar and comfortable with using these 2 terms. And again, it just takes practice. So a good way to practice this is to go back and explain, for example, the steps that are happening in the citric acid cycle, using the inputs and outputs, and just practice using those words. At any given point, if we've got NADH coming out, practice saying the NAD+ got reduced to make NADH, right? Or citrate got oxidized. So talk about which one is which. So now we've taken glucose, we've broken it down into 2 pyruvates, we've taken some of the electrons off, we've made a little bit of ATP, we've oxidized that into acetyl-CoA, making a little bit more NADH. We've sent that acetyl-CoA through the citric acid cycle, and in the process made a tiny bit more ATP, and we've made some more NADH molecules and some reduced FADH2 as well. We need to now do something to get a bunch of ATP, and that is going to happen using the high energy electron carriers that we've generated. Okay, so the last step is called oxidative phosphorylation. So in the presence of oxygen, we're going to phosphorylate something. What are we going to phosphorylate? ADP. This happens in or on the inner membrane of the mitochondrion. So if you look at this cartoon or this scanning electron micrograph, these folds are all membrane surface that's protruding into this inner space called the matrix of the mitochondrion. And the proteins that do these reactions are all scattered within and embedded through these membrane folds. All right, so we've got this relationship between structure and function now. We've got folding of the membrane, which increases its surface area and gives more places for these reactions to happen, because the more space we have to do this, the more ATP we can make. So this is the electron transport chain. And again, this bottom membrane is the inner mitochondrial membrane. If we go back a minute, there's an outer membrane as well. So this organelle is enclosed by two sets of membranes. There's one that makes the boundary between the mitochondrion and the cytosol around it. So that's the one that's colored gray here. And then there's a second set of membranes that make all these folds that are colored pink in this diagram, the ones that are protruding inside on the left. This is the inner membrane. So here's the inner membrane in this cartoon, and here's the outer membrane. And we've got the space between these two called the intermembrane space, the space between the membranes. Both sets of membranes are just like you're used to seeing with the plasma membrane. They're selectively permeable, right? Ions cannot diffuse through these. The only way to send an ion, like a hydrogen ion, through this membrane is to do so via facilitated diffusion or active transport. It needs something to physically move it across because it can't diffuse. Okay, so what are we going to do? What we want to do is harvest the energy in these electrons to do something. What they're going to do is work in the form of transport. So we've got this inner membrane, and all along the inner membrane are lots and lots and lots of copies of these proteins. And these proteins together form the electron transport chain. And their job is to transport electrons. So where do they get the electrons from? They either get them from NADH or FADH2, which came from either glycolysis, pyruvate oxidation, or the citric acid cycle. Right. I mentioned that NADH has higher energy electrons than FADH2, so they're going to start this process at slightly different points, but the end result is the same. Okay, so for each step in the oxidation reactions, when we're taking electrons off of things and sending them somewhere else, these are exergonic reactions, which means we've started with more energy than we end with. So what are we going to do with the energy released? We're going to use it to transport hydrogen ions, protons, from one side of the membrane. So this is called the mitochondrial matrix. If we go back a minute, this is all the space inside the mitochondrion, inside the inner membrane. It's the space that's colored kind of this orange color in this cartoon on the bottom right. Right, we're going to move them from the matrix to this space, this intermembrane space between the two membranes. So each of these giant proteins embedded in this membrane is actually a proton pump. It is an active transporter because we're going to move these hydrogen ions from a low concentration to a high concentration. We're going to get the energy to do that from these electrons that have all this energy stored in them. Okay, so your book names all of these molecules. You don't have to know the names. What I want you to do is pay attention to the pattern. All right, so we've got three major proton pumps, and they're fixed in place. They're these big, massive protein complexes. In between them, we have what are called mobile electron carriers, and these are like little electron shuttles. They can move laterally along this membrane, and so they can pick up an electron and then dump it off at the next stop. And then this one can pick up an electron and dump it off at the next stop. Every time an electron moves, something is getting reduced and something is getting oxidized. So if we start with NADH and its electrons, NADH gets oxidized, and this first complex gets reduced. So here's where the electron binds to it. So what are we left over with? NAD plus and a proton, a hydrogen ion. the energy in this electron, when it binds to this complex, is used to move a proton from one side of the membrane to the other. That takes energy, but we've given this complex energy, and so it's going to move this proton. That complex then gets oxidized, and this mobile electron carrier gets reduced. That mobile electron carrier then gets oxidized, and the second complex gets reduced, and it uses that energy. It's a little less than it had before, but it's still enough energy to move a proton from the matrix to the inner membrane space. Then the second mobile electron carrier picks up the electron, so it gets reduced, and this complex gets oxidized. It brings it over here to the third one. The carrier gets reduced, or gets oxidized. This next complex gets reduced, and it uses the remaining energy in the electron to move another proton into the intermembrane space. This electron is now out of usable energy to pump protons, but it's blocking up the chain in its position here. So one way to think of this is like a slide at a playground, right? If you go down the slide and then you sit on the bottom, then there's no room for another kid to go down the slide. And if they do, they're going to sit kind of halfway up the slide because you're still there. And if somebody else tries to go down the slide, they're going to sit at the top and not be able to move. And then nobody else can go down the slide, and everybody's mad at you. The same is true for the electron transport chain. So if this electron stays, then this electron is stuck here, and this electron is stuck here, and now we've stopped pumping protons, and we can't move any more electrons through, and your cell is ultimately not going to be able to make any more energy. So we need to take this electron off of the third complex so that this slide can keep moving. The molecule that does that is oxygen. Oxygen is going to interact with this electron. Oxygen is going to get reduced. The hydrogen ion is going to bind to it, so we're going to form H2O, and this is a byproduct. So the oxygen that you breathe, that you need to survive, right, that's critical for cellular respiration, doesn't come into play until the very last step. But without this step, this process stops. The process before it stops, the process before that stops, and your cells die. So this is why if you stop breathing, you won't live, right here. Okay, so electrons come through this way. If they start with NADH, they start with this purple molecule, they ultimately move to the green molecule, they move to the pink molecule, and in the process, they pump some protons. If the electron came from FADH2, it starts midway. It doesn't have as much energy, it can't start at the top, it starts partway down the hill, but it still pumps a couple protons because it's going to move to complex, this green complex here and this pinky orange complex here. So all the electrons are going to be, their carriers are going to get oxidized, and these electron carriers or these proton pumps are going to get reduced sequentially, such that that energy, electron's energy, is being used up in very small chunks, and it's being used up to pump these protons. We have not yet made any ATP. So here's the magical last step. There's an enzyme made up of a whole bunch of proteins, all organized together in a very precise three-dimensional shape. This enzyme is called ATP synthase. It's going to synthesize or build ATP. It needs energy to do that, right? That bond between the second and the third phosphate groups is very high energy. Where does that energy come from? It's potential energy stored in this proton gradient. So a few weeks ago when we talked about membrane transport and active transport, I said that ion gradients can be used to do work, and here's one of the examples. By moving all these hydrogen ions into this space, they're stuck, right? They can't diffuse through the outer membrane because it's not permeable to them. They can't diffuse through the inner membrane, the ATP synthase that has this nice little tunnel that allows them to diffuse down their concentration gradient, right? So this is kind of like facilitated diffusion. They're going to go from a high concentration to a low concentration. And in the process, the energy stored in that gradient is going to be converted into mechanical energy that spins this protein complex, and that gets converted into chemical energy because it allows that energy to be translated and make a bond between this phosphate group and ADP. And this process is called oxidative phosphorylation. We're going to make about 20 ATP for every glucose molecule we started with in this manner. So this is the bulk of the energy production in cellular respiration. every step we did previous was to put those high energy electrons onto carriers that would allow those carriers to pump protons using that energy so that ATP synthase could use the energy in that gradient to make a bunch of ATP. So here's our proton gradient. Here's ATP synthase. These protons diffuse through ATP synthase down their concentration gradient, causing it to spin, building ATP in the process. So here we have potential energy. We're turning it into mechanical energy, and that's being used kind of like a mill grinding flour, right? Is going to be used to build these chemical bonds to make ATP. And now we have energy, now we have ATP. So if we go back to our cellular diagram, we started with glucose. We broke it down into 2 pyruvates. We took a little bit of ATP out, a net production of 2, and made some high energy electron carriers. We then oxidized the pyruvate into acetyl-CoA, made a couple more electron carriers, broke that down in a series of reactions that produced a little bit of ATP in the citric acid cycle, but made many more electron carriers. And all those electron carriers got oxidized through oxidative phosphorylation. Their electrons were used to move hydrogen ions, protons, from one side of the membrane to the other. And then they diffuse through ATP synthase to make a bunch of ATP through oxidative phosphorylation. Right. So here's our net 2 ATP. Here's our net 2 ATP. Here's our anywhere from 26 to 28 ATP that came from the last step. And we're making about 32 molecules of ATP for each oxidized glucose molecule. And as I already mentioned, the role of oxygen is to allow, go back a minute, this electron to leave this last complex so that we can continue doing this process. In the absence of oxygen, everything backs up, everything stops. Right? It's when the roller coaster breaks down and everybody's in line and the line keeps getting longer and nobody is riding. Right. There are ways to do this without oxygen, not for humans, right? But there are some very specialized bacteria and organisms called archaeans that can live in the absence of oxygen. They use something else to take that electron off, right? They have evolved the ability to survive those conditions, right? So there are these weird examples that are really fun to study, especially because they have these useful applications for us where we've accidentally dumped or purposely dumped toxic chemicals into the environment. They need them to do this respiration so they can help absorb them from the environment. So this is not to make you more confused. This is to point out that, yes, there are ways for some organisms to live in the absence of oxygen. They still do these processes. They've just evolved the ability to have a different electron acceptor at the end of their electron transport chain.